Hello, everyone. Good morning, good evening, good afternoon, wherever you may be. My name is Sarah Jane Demy, and I am the founder and CEO of Demy-Colton. And it is truly my pleasure to welcome you here to the 17th installment of the Demy-Colton Virtual Salon Series. Today's salon will focus on sepsis, a really very important topic and one that has not gotten enough attention. As some of you know, Demy-Colton's mission is all about convening life science industry leaders to catalyze change. We know that we help drive innovation when we bring individuals together who are united by a shared passion and a common cause. And in our industry, that cause and passion is bringing therapeutics and cures to people in need. Now, at this time last year, we were about to launch a new meeting here in New York City called BioFuture, a meeting that's focused on the mashup that is currently underway between biopharma, digital medicine, big data, AI, healthcare, and payers that we believe will change healthcare as we know it. Then COVID hit, and it changed everything about what we are doing, and I'm sure it changed everything about what you're doing as well. So in lieu of BioFuture 2020, we launched the Demy-Colton Virtual Salon Series to continue our mission of bringing great content and great people together to inform everyone. We know the Virtual Salon Series will be here to stay even after we get back to our new normal, whatever the new normal is. We ask you to check our website for the upcoming series of salons and topics and speakers. It evolves constantly. And of course, I encourage you to suggest topics for us to cover. And I encourage you to join us at BioFuture 2021, which will be a hybrid meeting taking place in New York City, October 5 and 6. Now, before we get underway, I'd like to acknowledge some very important people and organizations that are helping to make this happen, our sponsors. I'd like to thank WBB Securities for the sponsor of today's salon, as well as all of our other virtual salon series sponsors. And I'd like to also acknowledge and thank BioFuture sponsors for their ongoing and continuing support of what we're doing. A quick word about the structure of today's program. We will have our conversation for about 55 minutes. There is a lot of content to cover, and I've got really great panelists. And then we will break and go into a 15-minute Q&A period. I think you should see a slide coming up that will show you where you can go and ask your questions in the pane below your screen. I do ask everybody to make your questions as succinct as possible, which will help us address them. Please note, too, that this salon, as with all our other salons, will be available online within a few hours after today's event, and it will be there for quite a while. Thank you for that part. Now let me move on to introducing our discussion. We're focused today on sepsis and the fact that it's a critical healthcare burden that needs rapid diagnoses to improve patient outcomes. But I think by the time we get through our discussion, you will understand that it probably needs much more attention than we're even giving it today. The way we came about organizing this particular salon is because I had a conversation with someone who had a conversation with somebody else, and we soon learned that there were, of the panelists that we have today, 4 of us have had sepsis, and in various degrees of severity, and some being addressed better than others. We're all here to tell the story about it, but as you'll hear from Tom, that's not true for many hundreds of thousands of people around the world. And I think somebody mentioned just before we got on that 11 million people die annually from sepsis. That shouldn't be. So, as I said, I'm a survivor of sepsis, and I really consider myself quite fortunate. Mine wasn't as severe as others. We need to change the paradigm. And as you'll hear again, some of the solutions that we use to address sepsis today haven't changed in years and years and years. And considering all of the technology that we currently have, that shouldn't be. So we will have a call to action when we get to the end of it. Now, it is my great pleasure to introduce my panels, my esteemed panelists. The first is Rick Wright. Rick is a senior vice president of pandemic prevention and response at the Rockefeller Foundation. And prior to joining Rockefeller, Rick led BARDA, the U.S. Biomedical Advanced Research and Development Authority. And as you will learn, he is an expert in pandemic response and preparation, among other things, Rick. Next is Prabhavati Fernandez, known as Prabha. She is the chair of the Scientific Advisory Board of the Global Antibiotic Research and Development Partnership. And she is chair as well of the National Biodefense Science Board. Prabha has a long and storied history as both a drug developer and a microbial expert, as you will see if you read her CV, which is available online. Next is Tom Heyman, and Tom is the president and CEO of the Sepsis Alliance, an organization that under his leadership is making great strides in changing the paradigm around sepsis in the US and indeed around the world. Tom will share some information with us, some statistics with us about sepsis that I think will shock everybody. And last, we're not to the last one yet. Sorry, Ciara. Ciara Kennedy is president and CEO of Amplix Pharmaceuticals, a company focused on microbial infections from fungus. Ciara also has deep experience as a drug developer, and furthermore, she did her PhD work in sepsis. And Ciara, you can clarify that, in a little bit. And last but not least is John Spurzel. He is the president and CEO of T2 Biosystems, a rapid diagnostics company that he purchased and took the helm of after his own recovery from sepsis. And he'll share a little bit more about his story. John has a long history of leading dynamic companies, many of which were in the diagnostics space. Welcome all. We are pleased that you could join us today, very pleased. Now, before we get into the questions, I'd like to go to each one of you and ask you why you're on this panel today. Rick, you got on a little bit late. You didn't hear that we were going to ask you that, but I'm sure you can explain why you're part of our discussion today.
Well, Sarah, thanks for having me. It's really a distinguished panel that I've learned a lot from in the past couple of years. I really became interested in sepsis, not only through my work at the U.S. government when we have been prioritizing some of the highest threats or worst threats that we face as a human population. And we recognize that bacterial infections and those that lead to sepsis are really deadly and causing significant death and costs in our public health. And I supported those programs. And to be honest with you, Sarah, it wasn't until I had an encounter myself with MRSA in a garden, everyday life, no thorns, just simple gardening and a nick on my thumb, that led to a MRSA infection and a disastrous experience I had with our healthcare system and recognizing what was happening and getting me the right treatment in a timely fashion that nearly led to the severing of my entire thumb or perhaps part of my arm because we didn't have the right tools in place for using them. So that ignited a passion in me to not only technically address sepsis, but really try to solve sepsis and join the universe and ecosystem and community of those who are trying to save lives by addressing this critical crisis.
Oh, thank you so much, Rick. Thank you. Thank you. Your story is really chilling as well. Prabha, may I turn to you?
Sure. Thank you. Thank you, Sarah. So good morning, everyone, or good afternoon. So my interest is, and I'm a microbiologist, and in a microbiology lab, we are trying to make a diagnosis. You get samples from the respiratory tract, urine samples, other samples, but the most important sample which comes in is a blood culture. And you need to get a response out back to the physician as fast as possible. Today, we have rapid methods which help you with those diagnoses. And we try to report it out to the physician as soon as possible. In drug development, we develop drugs for respiratory tract, urinary tract. There is no separate drug development for sepsis because the bug comes in from the respiratory tract, the urinary tract, the GI tract, or the skin. And so you develop drugs for those, and those are what gets used. And that's what I've been doing my whole life, been developing antibiotics for over 30 years. Now, what brings me to be of great interest is that the number of deaths, those of us who are on this panel are the lucky ones. We suffer, but we are still the lucky ones. I suffered over 50 years ago with salmonella, which is from the gastrointestinal tract, a foodborne disease. And I got treated. I survived. Was hard, was very hard. I survived. I lost 6 months of my college work. Now, interestingly, the way I was diagnosed is exactly the same as most hospital labs throughout the world, including the United States, diagnose salmonella infection septicemia today. And that's the sad part. We have rapid methods today, but do we really use them all over just because it's expensive? Staying in the hospital for weeks is very expensive.
It chills me when I hear you say that. So thank you, Prabha. Tom?
Yeah, thank you, Sarah. I'm here representing the patient voice, of which we see from our panel alone are many. Sepsis Alliance was founded, like so many organizations who do good work in the public space, not out of a whim, but out of a tragedy. And in our case, it was Dr. Carl Flatley who got the unfortunate call to serve. Dr. Flatley was practicing oral surgery in Tampa, Florida, had never heard the word sepsis before. This was in 2002. And it wasn't until sepsis took one of their family members, their daughter Erin, who was 23 years old at the time, perfectly healthy young woman, just graduated college, was going to be a school teacher, the perfect young adult ready to take on her life. And she went into her local hospital for a hemorrhoidectomy, simple oxymoron, perhaps, outpatient surgery. And she was sent home without any antibiotics, any follow-up care, any transitional follow-up. And she got worse, not better. And it became clear after the fact that she had developed an infection during that surgical process. The medical establishment at that hospital was not sepsis ready. They were not trained. They were not educated. As a result, Erin died 2 days later of sepsis, which led Dr. Flatley on his effort to make sure no other family would suffer that kind of just horrific, unimaginable tragedy. And when he started his work, he realized that there was no one doing anything in the United States about sepsis. There was no advocacy. The CDC did not have it on their website, was not doing anything about it. It was just not on the radar, even though the numbers were there. And in fact, Microsoft did not have it as a word. So if you tried to type in sepsis on your computer, it would get rejected. There was no sepsis. It didn't exist, according to Microsoft. So we've come a long way. At that point in time, only 19% of the country had heard the word sepsis, and most of them couldn't tell you what it was. It was confusion with septic tank, antiseptic. We're at 71% now. We are working hard to educate the public. This is largely a community-acquired syndrome, UTIs, spider bites, et cetera. About 80% plus start in the community, but many start in hospital and in healthcare as well. So we're educating the public through our website, sepsis.org. It's about 2.5 million visits per year. With the help of BARDA, we're educating healthcare providers across the continuum of care. We've trained over 40,000 providers to be as knowledgeable about sepsis as they are about stroke. Sepsis is twice as common as stroke, twice as deadly as stroke. And we're also working in the policy realm. So we're really, in the work we're doing here, I feel like I'm in a grand jury and we're deciding to indict sepsis. And, this has to be done. And with the work in diagnostics, either we prevent the infections before we identify and treat them early, and that requires diagnostics. There's so much guesswork in the process of diagnosing and treating sepsis that it really is difficult on healthcare providers who are caught in a vice between treating early and the pressures of antimicrobial stewardship and resistance. So the solution is prevention and better diagnostic tools and attention to antimicrobial resistance. Thanks so much.
Thank you. Thank you, Tom. We're going to get to a call to action at the end of this as well to reinforce all of these messages that are so important. Ciara.
Hi, Sarah. Thank you very much, first of all, for shining a light on this really important topic. I feel for me personally, this is almost like me coming full circle. As you mentioned, my PhD work was in sepsis and the inflammation, the inflammatory immune response side of the sepsis continuum. And today I am driving forward a company that is bringing innovation into this space, which is challenging in the current financing market for these types of products, these types of companies. But patients deserve better solutions, better treatment, safer drugs, ways to prevent sepsis by addressing the infection up front. You know, I'm fortunate I don't have a patient story, but I'm unfortunate in terms of my PhD supervisor, right about 6 months after I completed my PhD, died from sepsis after spending her life studying it, researching it, trying to understand the immune cascade that follows. And, you know, the irony and the tragedy of that was with me today, and something that motivates me to get better options there into the clinic, out to patients in any way we can.
Okay, we're here to help get that message out there with this particular salon, and we'll keep at it.
Thank you.
John, can you hear us?
I can.
Oh, okay. Tell us your story, John. Why are you here?
Me, and like the other panelists, thank you for shining a light on sepsis. The human and economic toll related to sepsis is staggering. The numbers, as we'll hear today, are enormous. I'm here for two reasons. As the leader of a diagnostic company that's focused on rapid detection of these sepsis-causing pathogens, I see an enormous opportunity to change a very long-standing standard of care that's failing on so many levels. I also have a personal story. In 2017, I was diagnosed with one of the world's rarest and unfortunately deadliest disorders. I spent 55 days in a cardiac surgical intensive care unit, endured numerous surgeries, was on life support, and ultimately received a life-saving heart transplant. Post-transplant, as is often the case, I obtained several multi-drug resistant bacterial infections, one of which became septic. So the second reason I'm here is to deliver on a promise that I made in honor of my heart donor to pay it forward in a meaningful way. So thanks for having me.
Oh, thank you. Thank you so much. Thank you all for being a part of this, this really important discussion. Before we begin, I think we need to define sepsis. I think people hear the word, you hear septicemia, you hear septic shock. Is it a disease? One of our questioners said, you know, isn't there a drug for sepsis, as though we're one disease? So I would like to turn to both Tom and Prabha, and we'll get to everybody on this as well. What is sepsis? It's not a disease, it's a syndrome. It's a response. Am I right? Tom?
We don't care what you call it, as long as you call it. So
Okay.
we had this debate inside, and it led nowhere. So I don't care what you call it, as long as you call it. And you know, we speak to healthcare providers. We also speak to the general public. And you know, I continue to hear doctors on television news calling it a really bad infection. It's not a really bad infection. It's your body's dysregulated response to any kind of infection, bacterial, viral, parasitic, or fungal. And, you know, now with COVID-19, there's an opportunity to shine a light on COVID-19 is a viral infection. Bad COVID-19 is viral sepsis, which can also lead to bacterial sepsis. So a lot of times I've stolen this, but we like to tell people it's kind of like a bee sting. When you get stung by a bee, it really hurts. but it's not necessarily a problem unless your body has that anaphylactic response, like Sierra was talking about, the immune response to that bee sting, when you can have real problems. And similarly with sepsis, it begins with an infection, but an infection unto itself is not necessarily a problem unless your body has that reaction to it. So we've found that that's a very helpful way to describe it to people.
Okay, Prabha, do you want to add to that?
Sure, thank you. So, there's the organisms which are causing the bloodstream infection, which then could result in septic shock, which Tom just spoke about. They come from the portals of your body, which is the respiratory tract, the urinary tract, the GI tract, and the skin. So when we develop drugs, we develop for those infections in those portal areas. So when someone asks the question, don't we have drugs for sepsis? Yes, we do. We have new drugs for sepsis, new drugs which perhaps address almost all of those drug-resistant strains we talk about. The question is, when you have the sepsis, can you find which organism is in there? Because it is usually one or perhaps two. There isn't usually many organisms in there. There are many organisms in these other body sites, but what gets into the blood is that most virulent one from that infection at those sites. So now how you diagnose it, but you need to know what is that appropriate therapy. And the important thing is you have to add it quickly, but you may not have the diagnostic result immediately. But once you know what it's susceptible to, you need to use the best therapeutic out there. And there are even something like ceftazidime avibactam, which is called Cas-Avi for short, would work against the majority of gram-negative infections. Vancomycin, even an old drug, daptomycin, would work, but you've got to know what that bug is.
You're getting us into our discussion. The next phase that we want to talk about is diagnosis. What is the problem with diagnosis right now? And I think, Prabha, let's pop over to Rick for a moment and talk about Rick's saga with being diagnosed properly.
Well, Sarah, thank you. Diagnosed properly is not exactly what's happening these days. So there isn't a proper diagnosis that's happening. It's amazing to me. From my role in BARDA, we invested in various technologies, new diagnostic assays, things that could detect a bacterial organism, characterize that organism, and actually tell you which drug could be effective. And some of those are already approved by the FDA and should be used in the marketplace today in hospital care settings. However, my experience just 2 years ago was very old-fashioned, very traditional. I mean, we relied on the culture assay. Matter of fact, when I went into an urgent care center with the signs and symptoms of an infection on my thumb, the standard of care wasn't even to culture it. I had to practically beg the healthcare provider to take the culture and to see if they can identify the organism. And then, of course, they gave me a general antibiotic, Bactrim, sent me home. It was less than 10 hours later, I was in the emergency room with that spreading. In the emergency room, they took a lot of blood in those large bottles that looked like a airline alcohol mini. They took several of those to culture and still never cultured that organism until 6 days later. I went back they gave me a different antibiotic, sent me home. I went back to the emergency room about 12 hours after that. They gave me a different antibiotic and then decided to admit me into the hospital. In the course of 3 days, I had 7 different antibiotics, none of them working, is why they scheduled the appointment to open the thumb or remove the thumb. Meanwhile, 6 days later, they got word from the urgent care that they did diagnose something from that culture. It was a MRSA. I was on an orthopedic ward in the hospital because of the location of the infection, walking the halls every day for 6 days, so could have infected lots of elderly folks there for hip and knee replacement. It's just a lack of recognition of this type of condition and the risk involved. So there are diagnostics today, Sarah. There are amazing diagnostics. We'll hear about some of them in this discussion, but they're not being used. We're still relying on a decades-old culture assay that may or may not work. And meanwhile, the doctors and healthcare providers are prescribing antibiotics and antifungals randomly almost, and driving resistance to these bacterial organisms even more.
Yeah, I think one of the topics that we'll get into is the institutional pushback, if you will, on updating systems. So we'll move into that. But let's jump over to John right now to talk about diagnostics.
Sure. Sarah, if I could just add a couple comments to what Rick said, because
Sure.
this is the antithesis of precision medicine, this current standard of care that we're using. We're relying on blood culture, which has two significant challenges associated with it. One, it's notoriously insensitive, as Rick said. How many cultures had to be done in order to detect the pathogen that he had? We can do better. The second is it just takes too long. Typical blood culture takes 1 to 5 days, 2 to 3 on average to turn positive. And unfortunately, it's a race against time because patients that are at risk of sepsis don't have time. For every hour of delayed appropriate treatment, the mortality rate increases by as much as 8%. So we don't have time with these patients. And that's why rapid diagnosis of these pathogens is so critical. I mentioned before that this current standard is failing on so many levels. The most important is just within the U.S. healthcare system, we spend $62 billion associated with this. We also lose 11 million people worldwide, including 270,000 people in the United States each year. The data shows that these broad spectrum antibiotic protocols are only optimal between 30 and 60% of cases. So if that's the best we could do, then so be it. We'd have to wait for the science and the technology to catch up. But it has caught up because of investments that have been made by many organizations, including BARDA, that Rick previously led. And these technologies are now available. So the real challenge is about adoption and making sure that these tests are used and making sure clinicians act on the results of these tests.
Yeah, I think that's a really important topic of discussion, and we'll get back to Prabha on that part of it. Ciara, can you speak about testing for fungal infections? I think you mentioned also that this is really behind.
It is. We've heard about the problems with blood culture in terms of bacterial infections. It's even more amplified when it comes to fungal infections because it takes even longer to get a positive blood culture and they are less sensitive. And depending on the fungal pathogen, you may not be able to access a site. It may not be circulating in the blood, but it could be invading through the wall of the heart or into the brain. I have heard way too many times that I care to from physicians that, we found out what we were treating from the autopsy report. That's just devastating. And I think that it is one, I guess, nuance between bacterial infections, as we've heard, these can happen randomly, very, unpredictably. The patients who typically get life-threatening fungal infections are a different type of patient. They are a patient that is dealing with an underlying cancer diagnosis or have had a solid organ transplant. So those are patients that our healthcare system is investing enormous resources into to address their underlying disease, whether it's, expensive chemotherapies or immunomodulatory therapies or transplant or stem cell transplant. And then they're on the cusp of recovery, on the cusp of remission and beating their cancer, and a fungal infection can take them out in a matter of days. That is devastating all around and not a way to protect our investment into the healthcare and treatment of patients we've made so much progress on the oncology side of the house, but we're losing ground on the antimicrobial side, and it really is a shocking waste of potential and resources at the same time.
And one of the other numbers, thank you, Ciara, that I think, Tom, you have on your website is that sepsis is also the number one cause of maternal mortality in the United States. Again, another shocking number. So.
Yes. Yeah, it's actually the number 3 cause of maternal death in this country. And we know that the rate of maternal death in the U.S. is going up, not down,
Right.
which is unique to us for developed countries. So it's a significant issue when it comes to maternal health.
Okay. What I'd like to do now is to shift into, if we can, a discussion about what the current, now that we talked about diagnoses and the fact that it's very difficult to diagnose right now, what will it take for rapid diagnostics to be made available in hospitals in the United States and worldwide? What will it take? I mean, John, I know you're in the business of that. Are there barriers to adoption?
Well, I would just reinforce that these products exist today. As I mentioned to you before we started, we have the only FDA-cleared products that are able to quickly detect these pathogens directly from whole blood. That means they're culture independent. We don't have to wait days for a positive blood culture.
Right.
It won't take several hours to detect these pathogens. We can do it directly from whole blood, and we have actionable information for the clinicians. So that's really important. There's a lack of awareness about this, quite frankly. This is a long-standing standard of care that has been in place for decades. So part of it is awareness, part of it is education, but then we're also going to need support from the government in terms of not just for T2, but for other companies as well, continued investment in these technologies to address the risk and the threat of antimicrobial resistance. That is a serious looming problem that we have to address. And we can't keep throwing antibiotics at patients when we're not sure what pathogen they're afflicted by.
Right. Great point. Great point. Prabha, when we spoke 2 days ago, we spoke about the treatment protocol that is widely accepted at hospitals and which has not changed. Can you speak to that and talk about the problems with that?
Yeah, so the treatment has been if a patient is suspected of having sepsis or septic shock, patients are given broad spectrum antibiotics, but the number of deaths was increasing rapidly. Each year on year, it was increasing. So in 2015, CMS passed a new rule called use the SEP1 bundle. So this sepsis Emergency Management 1 bundle. So when a patient comes in with sepsis or with fever, with a suspected bloodstream infection, the doctor has a certain protocol to follow, and he or she has to follow every one of them, including starting an antibiotic, a broad spectrum antibiotic, within the first 3 hours, preferably within the first hour. If those protocols are not followed, there is penalties in that the entire payment for the bundle will not be paid. So the blood culture, the clinical chemistry, all of the diagnosis which is used for the patient. So the problem is, it's a good thing that the CMS did this. You get treated within the first hour. That's great. You get a broad spectrum antibiotic to cover everything. That's good. But then what happens to all those diagnostics you've done? The results come back in a day or less, sometimes in 2 days, but the physician has just given you a broad spectrum antibiotic to the patient. That is not looked at. So this way you must adjust the treatment to suit the bug. As Rick just mentioned, it could be a MRSA. Change the antibiotic right away to suit that bug with that susceptibility. As was just mentioned by John, the technologies are available today to know which bug is it, what is the susceptibility, either de-escalate the treatment or escalate the treatment. Whatever is needed is available. There are 3 or 4 different gram-negative drugs are available today, many of them funded by BARDA when Rick was there. And these are excellent. They can be used. Cefiderocol is now approved. It can be used. It's from a Japanese company called Shionogi. It can be used. However, it's kept on the shelf for the reserve for those resistant pathogens. And those diagnostics tells the doctor what to use. And that's what we should encourage for CMS to encourage the use, not just of doing the test, which is what it's paid for now. Every physician does the test, but to read the test And then if the reward is for reading the test and doing what the test calls for, that's where the emphasis should be.
Yeah, so I think we have a situation, and all of you can correct me if I'm wrong about this or off target, where someone gets infected, someone you don't know immediately whether or not your infection is septic. Your doctor doesn't know if you bring it to your doctor's attention. For me, I called my doctor because the pain that I had was extraordinary beyond, the pain that I had from my broken ankle. People may complain of not feeling well, and sometimes they're just simply not diagnosed. Doctors don't pick it up potentially as sepsis, which is where Tom's communication and awareness campaign about sepsis is so critically important. But then you have, and probably I think we spoke about it, Ciara, we spoke about this, an institutional situation where old line treatments are prescribed, the old standard of care is prescribed, where in some situations doctors would not be reimbursed or hospitals would not be reimbursed if they took the more expensive therapeutic that may be appropriate. So you have slow diagnoses that are missed because if you treat with the broad spectrum, it's over and done with. You think that you've taken care of everything. And you have, the institutional resistance to change. I know, Ciara, do you want to address that?
Yeah, and I think it builds on the concept that Prabha just introduced about the bundle payments. Mm-hmm. So the bundle payments for specific disease codes are how hospitals get paid. So, I think the cold truth is if a hospital needs to use a branded drug, a branded antibiotic, they're going to lose money on that bundle payment. So generic drugs are first line. And there are certainly cases where a generic drug is appropriate and sufficient. But I'm talking about in the cases where they're not. So patients are treated with generic drugs, not necessarily the absolute best drug for that patient at that time. And it may be because the newer drugs are not yet on formulary, which can take an extended period of time in this sector. It's not true for other sectors. In oncology, for example, new drugs get onto formulary much more rapidly, and physicians can use them. So there is just enormous budgetary and financial constraints and considerations that are driving prescribing practices of antibacterial drugs. And you're talking about the difference between, a couple of dollars a day versus $1,000 a day. That's a big number. And it's a big number under a payment that's capped and bundled. So we have been doing a lot of work over the last number of years to raise awareness and education on this fact and to promote uncoupling of reimbursement from this bundled code for antimicrobial products. There's this concept of, in our industry, in the drug development industry, value is so closely tied to volume. So the more of the drug that gets used, the more return for investors. We need to break that mold and break that model in this case. We need to decouple value and volume of use of antimicrobial products. But if we don't have a system and a framework to deliver value, the innovation will not be funded. And, it's almost like that fire extinguisher that you have in your house that you bought, but you hope to never use. But you're happy to pay the price for it in case you need it someday. I think that there's a lot of different ways that we can change how these types of products are paid for and reimbursed that will make sure that A, they're available for patients, and B, they're used in the right patients at the right time. And the Disarm Act, Pasteur Act, these are all pieces of legislation that are being discussed to try and address this. I think there's a misconception, though, that these are sort of handouts to pharma, and it's the opposite. They really are a safety net for patients.
Yeah. How has COVID, and this is an open question for everyone. I I think COVID has probably changed the way that we look at bugs and infections from bugs and brought attention to the space. I think John or Tom or both of you mentioned that sepsis COVID is the number one killer of people that come down with COVID. Has that shined a new light on this space, and will it enable change to happen in this space, the type of change that we need in order to get away from 570,000, Americans dying every year from sepsis because of a combination of either not getting the access they need, lack of knowledge, institutional resistance? What will happen? I open that up to everybody. Put your hand up if you'd like to take that question first. John did it first, then Tom, then Ciara.
So COVID-19 and sepsis are inextricably linked. Critically ill COVID patients that are hospitalized often die of multiple organ failure, which is by nature the definition of sepsis. So there's a clinical link. We saw that clinical link and we saw that data early coming out of Wuhan, China, and we saw similar data in the United States. So there's a clear clinical link between critically ill COVID patients and the risk of bloodstream infections that can lead to sepsis and ultimately death if not treated appropriately. In terms of how we deal with that, we obviously have to pay special attention to critically ill COVID patients that are hospitalized to make sure that we treat them appropriately and they don't have a terrible outcome. And I think, as we said before, these tools exist to make sure that isn't the case.
Okay, thank you. Thank you, John. Tom?
Yeah, I think COVID has created, obviously devastation, but also opportunity in that, we have this new infection and we need to label it as an infection that is, wreaking havoc, and the havoc is mostly attributable to sepsis. So I think we have, an opportunity that we thought we had with Ebola, but fortunately we did not. With COVID-19, and when you think about this, I can imagine no more bipartisan issue than what we're talking about here. We're talking about saving lives and we're talking about saving money. So, I think we have a refreshed opportunity to approach this through things like Pasteur and Disarm, and to really take this battle forward. Why is sepsis the most expensive cost of acute care? Is it all at the front end, or is it that we're not doing things properly at the front end, which is causing tremendous length of stay? And 40% of patients who are lucky enough to survive sepsis are back in the hospital within 90 days. 2 out of 5 are back in within 90 days. So
I have to shake my head at how much damage there is, because I'm sorry to jump in on you like that, Tom, but how much damage there is because of how blind we seem to be to it.
We are a one-eyed monster when it comes to sepsis, and it is the devastation. There are 14,000 people who become amputees each year from sepsis. 14,000 people are losing limbs, and we are not thinking about this holistically. Payers are looking at the front end, and there's this huge back end of ongoing care that's required, whether it's cognitive, psychological, PT, OT, that is extremely, extremely expensive. And our hospital boards paying attention to the fact that 1 out of 3 of their patients who die are dying of sepsis. Do they even know that? So
do they even admit it? If they know
it, do they admit it? Are they getting those reports? And if so, can you unsee those reports? I don't know how you could.
I don't know. So, Ciara, I know you had your hand up. Then go to Rick, and then Prabha.
Okay. A couple of different points. We're absolutely seeing data about secondary infections to COVID, be then bacterial and fungal, but that's not news. We've seen this before. So it happens post-influenza. So the secondary infection post-influenza, that's been something that's been around for a long time. We've seen an enormous increase, and I'm going to come at this from an investment perspective, an enormous increase in interest in companies that are developing COVID therapies. But we have not seen an uptick in interest in companies developing new antibacterial or antifungal antifungal drugs. And so I think that there's a blindness and a blinkeredness going on where if a patient dies from sepsis or from, so let's just talk about COVID. It's top of mind. If a patient dies from a secondary infection or sepsis due to COVID, people think about that patient as they died from COVID. COVID is not what's killing them. It's the sequelae and the consequences that happen afterwards that are causing their death. But there's almost like a misidentification or a recognition of what is killing the patients. And we don't, if I mention cystic fibrosis or DMD and just devastating diseases, but we can almost in our minds picture what that patient looks like. You say sepsis, and you don't get that same clear picture. And I think that until we can show the world what that picture is, it's just harder for people to connect to it. And, I think that we see more and more antimicrobial companies that have gone through bankruptcy in the last couple of years after they've done all the heavy lifting of getting a drug approved. That's the time when good things should be happening, but instead these companies are going bankrupt, and multiple companies that have less than 6 months of cash. So potentially innovative new drugs that could help these patients may never make it because of the funding environment. And that hasn't really changed in COVID times for those companies that are focused on the antimicrobial end of the spectrum.
Thank you, Ciara. Rick.
And those are all very important. There's a lot to unpack in all the comments already. I also want to put on the table that, the question is, do the patients really fully recover? So either the COVID patient, we're learning more about the long-term effects, the long hauler effects. And it's very similar to what we've learned about sepsis patients. Believe it or not, Tom mentioned 40% of the patients come back. We are seeing evidence. We did a nice, thorough study with CMS a couple of years ago. Number one, not only highlighting the number that John threw out is $62 billion healthcare costs. That's just for CMS beneficiaries in the subpopulation. Doesn't even cover the entire population. But we're also finding that many of these patients never really fully recover. It almost creates a chronic condition after you had sepsis. Once you've had that serious bout with sepsis, you are much more susceptible for another bout of sepsis. And next time, it's actually much more severe and could likely lead to death. So the point that's been made, I think, here several times is we don't have to let it get to that point. There are so many things that we can do upstream to detect sepsis, to detect bacterial infections, and to characterize the organism and identify the proper drug. Those tools are not being utilized. Number one, we have to do a lot more education for the healthcare providers. As Tom laid out, we need to have algorithms in the systems. There are biomarkers that need to be incorporated in those early triage tests and that we can identify, a computer can identify sepsis much faster probably than a healthcare worker. And a computer can remind the healthcare worker to think sepsis. If you don't think sepsis, you don't stop sepsis. And then it just cascades downward from there. And then, of course, the concept of reimbursement. Again, this is an area that I worked on with BARDA, not only developing new drugs and new diagnostics, but working with CMS to come up with new models to reimburse appropriately for novel antibiotics, number one. When I was treated with 7 different antibiotics before they got to that branded antibiotic that worked like a charm, but they had to try six generic antibiotics on me first. And then I quickly got a note from my health insurance company that they weren't going to cover, they needed to have a conversation, they weren't going to cover that branded antibiotic. It made me really think carefully and have a conversation with CMS about how we reimburse these branded specific antibiotics for these cases. These are life-saving tools that save lives and reduce the healthcare cost overall, especially if we know about it and use them sooner. And then we carry that conversation into diagnostics. Diagnostics are not treated with any level of respect for the impact they have in reducing our healthcare costs and saving lives. They are thought of as low cost, low commodity, should be, a few dollars or maybe $50 or something, and not really respecting that they could save tens of thousands of dollars, if not hundreds of thousands of dollars in a life, if used appropriately and timely. And they can also reduce the overall impact of further driving antibiotic resistance. So there should be value-based pricing in that reimbursement for diagnostics. I was really encouraged by the relationship that we formed in BARDA with CMS to start tackling the failed marketplace for antibiotics, realizing that most of the companies that were developing new antibiotics and the companies that got them approved by the FDA were going bankrupt within 1 to 2 years. Those antibiotics were not on the marketplace at that point, not available. They would not have been available to save my life or treat my thumb during that situation. So CMS heard that cry, and CMS began evaluating the different rules and different reimbursement and stretching a bit. And I know they started a momentum where I could hope they're continuing to look at those options. At the same time, we're hoping that the new administration and the folks in CMS will look at this diagnostic challenge as well, because diagnostics need to be reimbursed. And that is how they'll get the credibility, the use, I believe, in the healthcare system. And then these bills, Disarm and Pasteur Act, I believe from the experience I have and the work I had done on this in BARDA, are absolutely critical to right-sizing and balancing that marketplace for new antibiotics and diagnostics and antifungals as well, to make sure that those companies that develop these new tools are stable, are able to gain a foothold in a marketplace, and able to wait out the lengthy period of time to get those drugs on the formulary in the hospitals, and to understand the real costs and value that they should be for reimbursement without going bankrupt in the meantime. So the Pasteur Act and the Disarm Act, both of those have advantages in them that will help stabilize the marketplace for these life-saving medicines until we can figure out the right pricing and reimbursement model.
Right. Thank you so much for that, Rick. It sounds like a map for the road forward for us all. So one thing I do want to mention is that we have close to 375 people that have registered for this event, and they come from all over the world. We have representation from South Asia, East Asia, from North Africa, South America, and throughout the United States. And we have a number of people that are representing companies that work in this space. So I think it's really important for everybody to be on the same page and to join together so that we can advance the work done here. And I'd like to know what the Rockefeller may do to help, Rick.
Well, Rockefeller Foundation has been investing in precision health and overall global health initiatives for many, many years. They are identifying the critical healthcare needs and gaps around the world, identifying partners and methods and frameworks so we can try to address and close some of these gaps to reduce the impact that we're seeing from a number of the significant healthcare causes around the world. One of those, of course, is antimicrobial resistance and bacterial infections, leading to sepsis, leading to other downward spiraling healthcare costs around the world. Trying to find the equity lens to ensure that we have collected the right data from around the world and are building a platform to share those data so we can identify a newly emerging threat or track a threat that is beginning to spread and make sure every country has access to the same information so they can prioritize within their country and within the region and global level so we can get in front of these threats as they emerge.
Okay, we have only literally a minute to wrap up, and what I'd like to do is to ask you all to as concisely as you can, express your call to action, what we should do next. And I'd like to turn it over to Prabha now.
Thank you. One of the things to understand is that COVID is a serious disease. So you consider the pneumonia there, you treat with broad spectrum antibiotics, you might culture, you might follow those patients up. They're not done accurately now because those samples are contaminated. However, what I really want to say is that every infection is serious. You saw the cases which were just presented. You heard about them. They were simple skin infections, simple bone, whatever. It becomes very serious. Every infection is serious until it goes away, because it can become serious. They should be properly diagnosed and properly treated. So consider that every infection is serious. That's my message.
Thank you. Thank you, Prabha. John?
Well, it's hard to follow that. I think it was very, very well said. It sounds very overwhelming to listen to the stories, to listen to the data, to listen to the results. I think there's reason for optimism, and I think there's room for hope. There have been a lot of investments in technology. I think we need to continue to invest in technology. We need to continue to invest in education and awareness. We are fortunate to receive a very large product development grant from BARDA under Rick's leadership. We're grateful for that. And that has the potential for us to develop a very comprehensive sepsis panel that looks at bacterial, fungal, and resistance markers all in one test, and detecting up to 99% of the pathogens that we might see in a hospital in less than 3 hours. So things like that, investments like that, give us a lot of reason for hope and potential to change this, let's call it unsuccessful standard of care that we're living in today.
Right. It's really hard to get your head around or arms around the fact that we are so backwards when it comes to treating this type of disease, this condition, considering all the great advances we have in other therapeutic areas. Tom?
Yeah, I think one of the things that's just missing from the conversation is data. We don't have the data that can really help advance policy, clinical practice, or innovation. We have silos of data all over the country and all over the world. There are sepsis silos of data in the Department of Defense, VA, different healthcare systems, different states. They are not talking to one another, which means we don't have the advantage to kind of bring them together to bring insights into what's happening. And so as a result of that, Sepsis Alliance has created a new initiative called National Sepsis Registry Initiative, and we have a website, sepsisregistry.org. And among other things, we're advancing legislation for the creation of a national sepsis registry. And, we believe that like with the cancer registry, which was created back in the 90s, it was a game changer. When the registry became in existence for cancer, we saw incidence go down. We saw mortality go down. There was innovation went up. We have a similar opportunity here, we believe, to really change the game with data, which will allow us to educate and persuade and change hearts and minds and innovate more effectively as we develop, millions of records on longitudinal histories of what kind of person, what kind of comorbidity, what kind of diagnostic, what kind of treatment, what kind of outcome. Why does one person get sepsis and lose their limbs, and another person gets an infection and just walks away as if nothing happened? So this is one of our new areas of focus, and you'll be hearing a lot more about this initiative in the future.
Okay, great. We're supporting your initiatives.
Thank you.
Ciara?
So my ask and my call to action is literally a call. Call your elected officials and share your concerns about the ground we're losing in terms of antimicrobial resistance, in terms of if you have a sepsis story, if you have an infection story, share that with them. It really makes it personal. We do have some resources that make it easy for people to add their name and their voice to this push, and the fightagainst-AMR.com is one, the hashtag FAAR, so fight against antimicrobial resistance. These are all resources that make it very simple and easy for you to elevate and escalate this conversation in a way that we believe will be effective in supporting Disarm, PASTEUR, and possibly even other legislation that is needed to make sure that we don't revert back to the era where common curable infections are no longer curable and unfortunately will become more common.
Yeah, I have, okay. What we'd like to offer, Ciara, is the ability to post any resources on the website that will go along with the recording of this salon.
Perfect.
And of course, we've posted Tom's information. John, if you have anything, or Ciara, or Prabha, Rick, anything that you'd like to post, people can download those. Rick, you get the last word before we move to Q&A.
Wow, powerful. The reminder is sepsis is a killer. It's a significant killer of people every single year, and it's a very expensive killer, and it doesn't have to be. We can do something about it. The technologies are available today. Healthcare workers and hospitals and institutions need to start using it. Governments, federal level, state level, need to start reimbursing for its value in saving healthcare costs, Medicare costs, and its value in saving lives. And patients, individuals need to ask for it. It's amazing. I had to beg for it when I was infected from everyday instant life. This is happening to you and me. It doesn't have to have a special tragedy to happen. Everyday instance in life can lead to sepsis. So we need to be aware of it as individuals, and we need to, when we seek healthcare advice or guidance or treatment, we need to ask for it. We need to demand that we are treated with the most modern technology or diagnosed with the most modern technology, and not just let sepsis happen to us.
Right. You know, Rick, you bring up a really great point that we haven't gotten into, and we can maybe just end on it. You were knowledgeable. You knew enough to ask. You knew enough to demand. What happens when people don't have that same knowledge? I mean, it's really, this is why you have 570,000 deaths. If people don't know enough to ask, and that there should be better treatment simply because they haven't been exposed to it, that's a problem. We need that pull with the healthcare system to also ensure that we have change.
Sarah, can I just comment on that?
Absolutely.
Really critical point. Sepsis disproportionately affects the underserved. Not every patient can be the advocate that Rick was, or that I was, or that others were on this panel. Some people are on ventilators. They can't speak for themselves. These are critically ill patients that are going down very, very quickly. So it's incumbent on us to act on behalf of all the people that are at risk of sepsis, because they may not be able to act on their own behalf because they're in an underserved community, because they're critically ill. And I think that really needs to be underscored here.
Right. Thank you so much for that. Thank you so much, John. I mean, it gives me chills when I think about that. I know how I responded when I was in a healthcare crisis, and I'm knowledgeable. And it's only because of the business that I am that I know what I know. So thank you all. Thank you all for that. Thank you all for your calls to action. We're ending the formal portion a little bit later than we had anticipated. But that's because we had some, really, really great conversation with everybody. I formally want to thank each and every one of you for the work that you're doing in this space. And let's keep the dialogue going. Let's keep the pressure going so that we make change, in this area. It shouldn't be the way that it is, but it is. So with that said, we are going to move to our formal Q&A. And if you can all stay with us for another 12 minutes at least, we can get through some of the questions that we have from the audience. One of them is from someone here in the States who asks if we can talk a little bit more about the host inflammatory response in sepsis and the potential role of immunomodulatory therapeutics. Anybody want to, Prabha?
Yes. So we know, let's start with COVID, because there you see an inflammatory response when you have severe COVID. Everyone seems interested in that today for obvious reasons with the pandemic. So, the virus is not what is causing that very severe inflammatory response at the end. It's the sepsis the response to microbial products, which is causing that response. What happens is those products causes your complement to get activated, causes also coagulation to happen, and then what Rick had just mentioned, the long COVID systems which follow. This also happens also after other forms of sepsis. So it's nothing new, as Sierra has mentioned. We've seen this before. And the treatments which are available for complement activation, for those which stop blood clotting, are also available, and they're used together with the antibiotics to prevent the sepsis from happening. So these are all important products, which are not only, there are some old ones, but there are new ones which are being developed to take care of these situations.
Okay, thank you so much, Prabha. Does anybody else want to add to that?
I mean, I would add that, it's sort of like a runaway train that's gaining steam and speed as it barrels down the track. The sooner you can intervene and stop it, the sooner you are likely to have a better outcome. So, I think addressing the infection, that's great. But, we've seen a lot of anti-inflammatory, immunomodulatory drugs being tested in sepsis to mixed results, to be honest. They work for some patients, but there are a lot of patients where it doesn't work because that train's really hard to stop once it gains speed.
Right.
The immune system is so powerful and complicated that it can be challenging, and there are so many inbuilt redundancies within the immune system that targeting one specific cytokine, for example, may not be sufficient to kind of dampen that fire that's raging. So I really think that yes, we have to, and these interventions are routinely tried in sepsis patients, but typically not to great outcome, because it's the early intervention and the early aggressive action is what's going to change the outcomes in sepsis.
Right. Thank you so much, Ciara. We have another question. Number one,
Rick had a comment, Sarah. Sorry.
Oh, I'm sorry. I didn't see that. Rick, sorry.
I was just jumping in to. I mean, there's some work in this area, especially for COVID, in treating acute respiratory distress syndrome, or ARDS. And we can't catch every infection in the early days, and we can't get in front of that fast-moving train of inflammatory response. And however, there are still drugs, still treatments that can target the host inflammatory response or the damage that is being caused by that host inflammatory response, and can pull back people who are really close to death's door by as a consequence of ARDS or other downstream inflammatory outcomes. And we need to really prioritize the focus on some of those drugs. There are generally a smaller handful of companies that are developing those drugs, and it's generally a very difficult patient population to access and evaluate those in large numbers, thankfully. But we still need to invest in evaluating those drugs. We'll have those because there are, unfortunately, in COVID, but also in sepsis, patients that get to that stage. And these drugs can rescue them if they treat that downstream consequence of inflammation in ARDS. And I've seen the benefit of these in a handful of patients in a few drugs, and they're not getting the attention they need from government in terms of financial support. They're not getting the flashy limelight that some of the other rapid acting or very expensive monoclonals are that are treating the organism itself. So we need to make sure that these are not forgotten in our armamentarium and how we're going to address COVID and also sepsis downstream.
Wow, that one of our viewers asked specifically about ARDS ARDS. So you've just answered his question without me having to ask you the question. Moving on to another question, this viewer asks, is resistance to rapid diagnostics adaptation a fear of disrupting the existing hospital laboratory structure? And what is the care reimbursement structure for supporting rapid diagnostics? I think the first part is probably a yes, and the second part is maybe I don't know. Who wants to take that on?
So I can do a little bit if you wish, but shall I go?
Go ahead. Prabha, and then we'll pop back to Rick.
Many of the university hospitals, the large ones, will have the latest tools to be able to diagnose. But the large number of patients in the United States are going to smaller hospitals, peripheral hospitals. And there they are very, very worried about the cost. And they also send out samples for testing. So if I have something very serious, if that sample is tested, sent to a peripheral lab for testing, the results don't come back in time because it's being, mailed out. So that's not very good. Now, in the hospital lab, you do worry about space and time and cost. They're also under huge pressure for being within a certain budget for these bundled costs we talked about. So yes, cost is important. And so what we have to do is to impart onto that hospital and to the regulators that the cost of serious illness is far greater than that diagnostic equipment we might buy.
Right. It's like when you talk to people about medication, actually keep people out of the hospital and therefore saving a huge expense of being hospitalized. Rick?
I'll probably give an excellent answer. And I kind of liken some of that hospital real estate to things I've heard from emergency departments and other hospital laboratories when we've asked them the very clear question, why aren't they adopting some of these technologies? Some of the traditional technologies and the laboratory equipment takes a lot of space. And we liken it to the cereal aisle in the grocery store. I can have a new cereal that is magical and can extend your life 20 years and make you healthy and strong by eating a bowl a day. But I can't find shelf space on that aisle because it's dominated by Kellogg's and General Mills and Post and others. You get that one new drug, that new diagnostic, or that new piece of equipment in that limited space in the hospital or in the laboratory. It does mean sometimes pushing something else out. And that space is dominated by large multinational companies and equipment that have a lot of marketing power, a lot of other power to keep that equipment in place. And so how do you make room for a new technology, as Don described, the T2 technology, that can assess all of those things in a few hours, what we traditionally would wait 6 days on for some culture if you get a good culture assay? How do I bump out space to put that novel T2 technology in there that's going to save my life?
I would just add to what Rick said, getting real estate footprint, in this case in a microbiology lab, is always a challenge. There is limited space and they're very protective of that real estate. And one of the things that we did during the COVID pandemic was we introduced a molecular diagnostic test for COVID. We knew that was a critical need within US hospitals, and that got us into many microbiology labs that we weren't in before. And our plan is to subsequently transition those instruments to sepsis testing. So it was a creative way to try to overcome one of these big challenges. But we have to do more than that. We can't wait for an opportunity like that to get these technologies adopted. And I think it comes down, on the one hand, to making sure that we have the right clinical and economic data to drive changes in standard of care. But it's also going to take changes in treatment guidelines. It's going to take changes in reimbursement. And it's going to take continued investments in education, much like Tom was talking about, how just the awareness of sepsis in his time at Sepsis Alliance has increased dramatically. We're going to need to continue to make those kinds of investments if we want to change the standard of care.
Yeah, thank you for that. And does anybody else want to add to that? I have one final question.
Yeah, I would just echo John's comment about reimbursement. Margins in hospitals are thin. They're non-existent at the moment. I think so many hospital systems are losing money in this era of COVID. I think that reimbursement is a way to drive change, and particularly if it's appropriately structured.
Okay, thank you.
Can I make just one comment? There's also
yes, absolutely.
Yeah, there is, familiarity. Everyone is familiar with culture and susceptibility. You have to change a culture, not just the. So if it's okay, I can give a broad spectrum antibiotic and get away with it, but it's not okay, and that's the message we have to give. It's not okay just to give a broad spectrum antibiotic.
Right. That's a tough change to overcome. So, Tom, your work is increasingly more important to get the word out with all of us. So I do have one final question before we wrap up. And this gets back to one of the other comments that were made earlier about COVID and being more susceptible. Do you think there will be more genetic studies to see who was at risk of sepsis, given the large number of patients who developed viral sepsis from COVID this past year? Who wants to take that on?
I assume, given the importance, it'll be studied. I think the challenge is the number of variables that are likely to come to play, but I have no doubt that it will be studied.
Okay. Prabha?
Yeah, we do know that males are more susceptible. It's made our ladies as the stronger sex, so that was good. But certain androgen receptors, the Y chromosome is a bad thing. We know that now, and interferon is a very big link. So there is quite a bit of evidence coming out as to what may be there, but we still don't know. What is the link to hypertension? What is the link to obesity? Because we know certain populations are more at risk. We know the Neanderthal genes left over are a great risk for patients. Yes.
Wow. Okay, with that said, does anybody have a closing comment that you want to make? We are up at now 12:16. So we
want to thank all the people who are listening, and we want all your support to help.
Yeah.
I could not agree with Pava more. We need everybody's support to change this paradigm. So thank you to all of our listeners. Thank you so much. And from my point of view, thank you very much to all of my esteemed panelists. This was a great discussion. We know from our experience and our other conversations that we've had that this could go on for hour after hour after hour as we go down different pathways. So with that said, we are coming to the end of this virtual salon. I'd like to thank again WBB Securities and all of our virtual salon series sponsors and our Founding Bio Futures sponsors for their ongoing support of the virtual salon series. And again, we'd like to thank the audience, and invite you to take the brief survey at the end of this as you're signing off. And don't forget to join us for the next virtual salon, which is spectacular, shaping the way healthcare thinks about funding on April 14th. So I wish everybody happy holidays, healthy holidays, and I look forward to seeing all of you in person when we can meet in person. Thank you all.
Thank you, Sarah.
Thank you.
Thank you, Sarah.